Okay, hello everyone. So in this recording, I will guide you step by step to complete your your independent assignment. So previously I gave you a detailed instruction on how to complete your assignment. However, I received some reports from among of you that have problems with the softwares and also some of the problems with the license of the software. So in this tutorial, I will give you the step-by-step to use other softwares. Okay, so this one will be easier for you to use compared to the previous one. So hopefully this will allow you to complete your independent assignment on time. So before we start, I would like to just review a little bit about your independent assignment. So in this assignment, you are asked to optimize or find optimized ligands for inhibiting the histone deacetylase enzyme. So this enzyme basically is a class of enzyme that remove acetyl groups from lysine on a histone. So histone is a protein that is used to pack the DNA in chromosome. So by removing the acetyl groups, it will allow the histones to wrap the DNA more tightly. So because of this, the DNA expression will be inhibited. Okay, so overexpression of HDAC2, which is the abbreviation of this one, this enzyme is associated with prognosis in many cancer. However, the exact nature of those genetic defects are varied among different types of cancer, but usually it includes the deactivation of genes that code for programmed cell death or apoptosis. So therefore, because the removal of acetyl group cause the DNA to be packed more tightly, it will inhibit the expression of genes that code for programmed cell death. So in that case, it will promote the proliferation of the cancer cell, which we don't want that to happen, right? So therefore, we want to inhibit this enzyme over here. So then therefore, the DNA can be unpacked, and therefore the genes that are responsible for inhibiting the cell proliferation will be expressed. Okay, so this is just a little bit of an overview of the mechanism that I said before. So this is the cancer cell, and inside of the nucleus of the cancer cell, it contains the chromosomes, and the chromosome consists of DNA that are packed around the histone proteins, right? So some of the genes in the DNA code for suppression of the proliferation of the cell, right? So this expression can be switched on or switched off by the epigenetic marker, for example, like acetyl that are attached in the histone proteins, right? So for example, if the HDAC enzyme work, it will remove the acetyl around this histone, and therefore it will cause the DNA to be packed more tightly, and therefore it will inhibit the expression of the DNA. Okay, but another enzyme is responsible to attach the acetyls in the histone protein, and it will cause the unpack of the DNA. So therefore, the expression can be proceed. And because the chromatin are open, then the genes that are responsible for apoptosis will be expressed. But when there's HDAC, as I said before, it will close the chromatin so that the genes that are responsible for apoptosis will not be expressed. So that is the role of our enzyme over here. So therefore, in this independent assignment, you are asked to find inhibitors that can inhibit this enzyme. Okay, so we will use computer-aided drug design tools to aid for this pursuit. So in the assignment, I gave you a lead compound over here and then 5 sets of the analogs of the lead compound, which we will evaluate using the docking. So these are the analogs. So each of this, so this is the first set, the second set, the third set, the fourth set, and then the fifth set. And each set has been modified for the substituents. For example, in this one, the RL substituents are modified, this one for amide, this one for the chain length variance, and so on and so forth. Okay, so what we want to do is basically to compare which analogs have better has the best binding to the HDAC2 protein. Okay, and we will do that using the computer-aided drug design tools. So therefore, the objective of this assignment is to perform molecular docking and interpret the docking results of molecules designed to competitively inhibit an anti-cancer target called the histone deacetylase 2 or HDAC2. And the scope of this assignment will include performing target and ligand preparation, performing molecular docking, and also interpret the docking result. So some resources are used, including the UCSF Chimera. So this will be used instead of the previous softwares that I gave you before. So this one is easier to use. So hopefully this will help you to complete this assignment. And then we will use 2 databases, the PDB or Protein Data Bank, also the PubChem. So the PDB consists of information about protein structures, while PubChem for the ligand. So this contain chemical structures. And then we will also use SwissDoc to help you to perform the docking. And this is a free online web service that we can use to dock our protein with the ligand. Okay. So the only one that you need to install is only the UCSF Chimera. So I will show you the step by step later on. Okay, now what you can do is to go to this link over here. You can just go directly to it. I will show you. So if you go to this link, you can find different installers, right? So you need to go to the daily builds section. So in this section, new builds are made. So this can include the most updated version. So you can look at the date to see which one is the latest, and you can choose the installer based on the platform that you use. So since I use Microsoft Windows, so I can just click on this one, download it, and then just follow through the installation process, and then you can open the Chimera. Now I will open the Chimera. So after I install the Chimera, it will look like this, right? Except you will not have these tabs on the right. And what you can do is to fetch the protein information, okay? So if you click on that, click File and then Fetch ID, it will show like this, right? So you can fetch the protein structure from different databases, right? Or ligand structure or whatever structure that you want. So in that case, I will use the PDB. Okay, so I will just tick on that and I will fill in the ID of the molecule that I want to visualize. So how do I get the PDB, the PDB ID of the protein that I want to visualize? So I will just go to PDB, right? So the RCSB PDB homepage. And then I will just fill in the name of the protein that I want to look at. So in this case, we are looking for HDAC2 protein, right? And then click on that. Okay. And then you will find several structures of the HDAC2. The difference is just in the complex. So for example, this one is the crystal structure of human HDAC2 complex with an N2 aminophenylbenzamide. Okay, so I will just use this one, and then I will just remove this molecule later on. So I will show you how to do that, because what we want is just the structure of the protein, okay, not this one. So I will just put the code, the PDB code of this protein. So I will put it in our Chimera. So just put the code over here and then click fetch. Okay. Okay. So after I click fetch, the Chimera will download the information. So if you look at over here, it is downloading the information about HDAC2 protein. Okay, we will wait until it finished. So okay, now it works. So this is the structure of the HDAC2 protein that you want to look at, right? That you want to study. So over here, you can see that the structure contains 3 different chain, right? So if I click select over here, I can see that it has 3 chains: chain A, chain B, chain C. You can also look at this. So this is the glutamine monomer on the C chain, this one in the A chain, and this one is the B chain, right? I can also check the residues. So there are amino acids over here, and it also have several ions. So you have calcium, sodium, and zinc. So I can click on that. So it will highlight the location of the calcium ion. So look at that green balls. So they are the calcium ions. I can also check for other residues. So for example, zinc. So those are our zinc, right? This one. And if you look at this one and this one. So it also has several ligands. So the LLX and also the NHE are the ligands. So if you look at that, right? So that is the ligand, right? The LLX over there, right? So the LLX, and also it has other residue, the NHE, which is another ligand over there, right? So because we only need the structure of the protein, so later on we will need to remove that, okay? And it also has the solvent. So during the crystallization process, some of the solvents are also crystallized, and then we need to remove this as well. So this is the water molecule, right? So if you look at that, those are water molecules. So we will also need to remove that later on, okay? So the ions, we can just skip it because they are the cofactor of the enzyme. So we don't have to remove that, but for the ligands, the solvent, right, we need to remove that. And also because this is like the simple representation of the molecule, later on we will need to add the hydrogens, because later on the hydrogens can bind to the ligand, right? So we need to restore the hydrogens and also add the charges, right? Some of the amino acids are charged, so we need to add those charges. So now I will show you how to prepare this protein for docking. So how to prepare the target. So what we can do is go to tools and then structure editing, right? and then you can look at doc prep. So this feature over here will help you to prepare your molecule before performing the docking. So we'll just click on that. So we choose the 3-max, right, as our protein, and then we will choose some of the actions that we want to do with the protein. So for example, I want to delete the solvent. So this is just the default. So don't untake this one. So I want to delete the solvent, delete the non-complex ions. So because there are no non-complex ions in this structure, so I will just leave it blank. And then incomplete side chain. So if there are some structure that are incomplete, I want to replace it, for example, right? So, and then add hydrogen. So this is important. We want to add hydrogen and add charges. And I want to save the file later on to mol file, for example, but I don't want to save it for now. So I will just untick that. So click OK. Okay, so it's working now. So we'll just say OK over here. So it's processing and I will just click OK. It will add charges as well. And it already calculate the charges. For example, for zinc, it will be +2. For sodium, will be +1. For the calcium, will be +2. And then click OK. Okay. Okay, so there are some charges, some residues that cannot be calculated for the charges, but it will be just okay for this one. Okay, so it will be it will assign for 0. Okay. Okay, now we're done with the prep, and now we will continue to delete the ligands and also to delete the other subunits or the other chains. So I'll start with deleting the residues. So we have 2 ligands, LLX and NHE. So this is our LLX. I want to remove that. Click Action, Atoms or Bonds, and click Delete. So now they are gone. And I will do the same thing with our NHE. Okay, so that is our NHE. Action, Atoms or Bond, and then Delete. Okay. Now we don't have any ligand, only the protein with its cofactor, the ions, right? And now because we have 3 chains, we can dock this protein, but later on there will be just complicated because these chains are just the repetitions. So to have more efficiently perform the docking, we can just remove the other 2. So click chain and then remove. Click the chain first. So this is chain B, actions, atoms or bonds, delete. Now it's gone. Okay, I will do the same thing. Chain C, action, atoms, bond, delete. Now we only have 1 chain. Okay. Okay, I will just put focus on this. Okay, now it's in the center. So this is the protein that we want to target. So later on, we will dock our analogs, our molecules with this protein. Okay, we already add the hydrogen, we already add the charges, we already remove some of the chains, right? 2 other chains, and this is ready to be docked. What we can do is to save the file, click File, save it to PDB. Okay, I will save it in my folder, for example. Sorry. I will save it in my folder, right over there in teaching. I will save it in medicinal, so you can just find your own independent assignment folder right over there, and I will just put the file name as prepared underscore 3max. I will save it in PDF, a PDB file. Okay, and then just click save. Okay, now we're done preparing our protein. Now we will continue to prepare our ligand. So what we can do is to close the session first. Okay. Now we will start the same way by fetching by the ID. Okay. Now for here, because we don't have like the PubChem, right? PubChem ID over here. So what we can do is that we need to find the PubChem ID first. Okay. So we can go to PubChem. Okay, so if you go to PubChem over here, pubchem.ncbi.nlm.nih.gov, we can enter the name of the molecule, or we can put the smiles, we can put the formula, for example, right? But if you don't know, so we do have the structure over here, right? So we can also draw it. Okay, so what we can do is we can draw it using draw structures, right? And I can draw the molecule like this. Oh, sorry. Okay, so this is the same compound as this one, right? So I don't put the hydrogens, but if you want to put the hydrogens in here, it will be okay. Okay. So this is the structure, and what we will do is to search for the structure. Right. So this is the structure, the 2-aminobenzamide. Okay. Okay, what we can do is now we have the code that we can put to our Chimera. Okay, put the PubChem ID and then click fetch. Okay, so this is your compound. Okay. So let's check for it. Is it the same one with our? Okay, it's a little bit different, so we might want to search. So the program kind of removed Okay, now we will search the structure again. Okay, so by looking at this, it seems like you don't have the same structure in the PubChem. So if you have it, then you can just search. But because you don't have it, you need to draw it. So what I will do, okay, so previously we already tried to find in PubChem the same structure that we have over here, but it seems that we don't have the exact structure that has that is the same with our lead compound. So what we will need to do is we need to input the smiles. Okay, so we can draw it. If you're unsure about the smiles, then you can just draw it and it will find the smile for you. Okay, over here you can just put NH2, right? And then use this one, this one. Oh, sorry for that. And then oxygen, and then NH. Okay, so this is your structure. And what you can do is that you can get the smiles over here, right? So we can look at the right. If you don't have it, then what you can do is okay, you can build structure from smiles. Okay, so what you can do is that you can get these smiles. So this is the smiles of this compound. Copy, put the smiles over here, and then put the residue name. For example, I will put lead. Okay, apply. Okay, so now you have this structure over here. So this is your lead compound, right? So we will need to do some process again, just the same with the protein. Tools, structure editing, doc prep. Okay, just click OK, 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 OK. So it already this already ready to be docked. Okay, so this is your compound, and I will save it as mol too. Okay, so I will put it in my folder, the independent assignment folder, prepared lead, right? And then click save. Okay, now our files are ready